I'm pleased to be presenting the second lecture that Inder's going to be giving on the suppressor genes involved in cancer. And the reason why is because I think this story is going to show how Inder, I've learned this just recently because I was reading your papers on the BRCA1 gene, that shows your fearlessness as a scientist, I think. And because it was a big controversy for a long time what exactly those proteins were doing, BRCA1 and 2. And Inder's lab had some data that was different from the mainstream, but he stood by it, I think. Do I have this right? Yeah, okay. And, okay, I was hoping I wasn't lying. But anyways, and he stood by it, and it came out, and I think, there's some consensus in the field now. But that gene has had a lot of controversy over the years. And so anyways, so yesterday Ron gave a very elegant introduction, and Rusty and I were sitting there going, oh man, Ron's gonna read a poem, Ron's gonna, whatever. And so, and then Ron told some touching stories, of the last 39 years, of the multi-dimensional relationship he has had with Inder and some of the other guys. And, they played tennis, they went to exotic countries together, and things like that. And so I sat around this morning and I said, well, what did I do with Inder in the last 29 years? And you're going to be surprised. All right, let me put this down. We went to Irvine once. Okay. Of course, I was a lot faster than Inder. He was coming along behind me here. I don't have any pictures documenting it the way Ron did, so my daughter made a bitmoji of Inder and one of me, and then here we are. Okay, so that was our big trip together, Inder. Okay, that's my whole story, I think. Anyways, all right. All right. I wrote a haiku because I can't really do a whole poem, but I thought I could do a haiku to Inder. There once was a fellow named Inder whose enthusiasm you couldn't hinder. He stood by his guns on BRCA1 and refused to give up or surrender. So Inder, go for it. I'll leave you the poem.
Okay. This is me.
And Rusty, the pressure is on. Okay.
Thank you. Thank you, Joanne. I thought we went together to the desert once, but maybe you forgot. It's nice to see some people still show up. Otherwise, it's always tough on the second one. You feel like a man who went to a therapist. After an hour, the therapist said, you're crazy. The guy said, I don't believe you. I need second opinion. He said, okay, you're also ugly. So I'm glad some of you are here. So what I'd like to today talk is really, as Joanne mentioned, is on the BRCA1. There was a French physician in 1860 who was a Huguenot working in a clinic, a surgeon who often had patients. His wife was also a daughter of a doctor. And there were lots of people in that family of his wife, not his, who had cancers. So he started at that time, and this is now we are talking of 1860, he started documenting the people who were, these black circles are those in the family who had cancer, and many of them have actually breast cancer. And this led him to make this comment. Now think about it, it's in 1866. Mendel's law had only come in 1863, which were not really available to people till 1890, when Bateson really began to understand them and explain to people. And look what he says. He says, a person in whom are assembled unknown conditions which expose him to later become cancerous can transmit to his posterity the group of conditions before they have been manifested themselves in him. Also, having received the diathesis, which is a kind of the disease, from one of his forebears, he may escape during a long and healthy life the consequences of that hereditary state and may pass on to his children the disease from which he was spared. This is really quite remarkable, just the sheer power of observation that this was a carrier. Of course, he didn't know at that time, but that's the case. Very shortly after that, de Bovary, who probably certainly knew about Mendel's law because they were already becoming popular in and in fact Hugo de Vries had already started talking about them quite publicly, he said that, let me add a consideration of the inheritance of tumors. In order that a tumor may arise in such cases, the homologous elements in both series of chromosomes must be weakened in the same way. That begins to start thinking of suppressor genes, that you need two genes to be weakened in order for a tumor to progress. This was followed by very interesting experiments by Henry Harris. He was actually a Russian Jew who lived in Australia, then came to Oxford, and I actually happened to meet him in one of my talks in 1985. What impressed me the most, he was a Regius professor. He has this beautiful office lined up with books, like a masterpiece theater kind of stuff. And the most impressive to me was outside his window was the cricket club of Oxford. So it was pretty impressive. And he did a very interesting experiment. He made heterokaryons. He took cells from HeLa or some other tumors, fused them with the normal cells, and showed that the normal cells always were dominant. Cancer was recessive in this case. So that was it was never really done well, because when you make heterokaryons, it's hard to really count the number of chromosomes, but he was able to show that. But the real interesting stuff began to come when Ed Knudsen came up with the idea of two-hit hypothesis, which was really the retinoblastoma. Children who were born on one side eventually get on the other, but if you operate on one side, it never went. And you can see mathematically he did that there were two curves in the case of the two-hit and one single curve in the case of the one-hit. And that really was then substantiated by Webb Caveney and his colleagues to show there really was one gene which is the germline and a second mutation, and that led eventually Bob Weinberg and colleagues to clone the RB gene. Now, in the same time, there was a general sense that although the tumors are largely sporadic, I would say 80 to 90% tumors are sporadic, that is to say we acquired in our lifetime, but there is a certain amount and a number of tumors which are familial or have hereditary components to them. And one of them has been breast cancer. About 5 to 10% of all breast cancers have a component from the familial or the hereditary one, where majority of them are otherwise quite sporadic. In 1990, Mary Claire King, she was actually a mathematician originally, then went on to study biology with Al Wilson to understand evolution, found that there is a gene on chromosome 17 which is responsible for this hereditary or a familial type of a breast cancer. And then very shortly, there was this competition. Now remember, 1990 isn't that long ago, but there were no easy ways to clone, to do this long sequencing. The genome sequence hasn't been done as yet. So people were going chromosome jumping. People are making large numbers. It was about a 2 million base pair, what she came up with. But a company, Myriad Genetics, then quickly sequenced it. And in 1994, they came up with the sequence of the first of those genes called BRCA1, and 2 years later with BRCA2. These two genes are now about 5 to 10% of all hereditary breast cancers, and I'll talk a little bit more about that in a moment. The amazing part of these genes are the high propensity of causing the disease. If there's a mutation, and the mutations are very often in Ashkenazi Jewish families, that's how it was originally discovered in those cases, that by the age of 50, there's a 50% chance of getting a tumor if you have a mutation in BRCA1, compared to only 2% in the case of the rest of the population, which is sporadic. By the time the age is about 70, it's almost 100%. 90% people will have a breast cancer. And these are the worst kind. They are triple negative, and I'll show in a moment. They have a very bad pathology to them. But of course, in many cases, the BRCA1 also leads to ovarian cancer. In general population, there's about 1 in 8. About 12% of the women in the United States will have a breast cancer, which is mostly sporadic. And there are all types of them. In fact, if you look at the histology, because breast has so many different cell types, the pathology, the histology of different cell types is there. The basal type, the luminal type, all kinds of different types. And sometimes, even if the histology is very similar, the outcome of the disease isn't the same with the same treatment, which works on one or not on the other one. In fact, 50% of the sporadic cancers now look like have also a BRCA1 mutation during that some period of time. These are the triple negative. By triple negative, you mean there are two hormones, which is estrogen and progesterone. They are deleted for that. They are normally involved in the growth of the cells. And a HER2 receptor, which is a kind of a growth factor receptor to which Herceptin binds to it. So those are triple negative. They have a mutation in BRCA1 most of the time. And they are very undifferentiated. They have some basal-like properties, not totally clear. And one can, of course, Jeff can tell you a lot more about from his work on single-cell analysis what precisely the mutations are. So this is a pretty bad disease. And there are many interesting questions about it scientifically. The first question that we got involved in it, the way I got involved in it, was since you heard me yesterday, all our work was on oncogene FOS, JUN, REL, NF-kappa B. They were all transcription factors. And they were all dominant oncogenes. That is to say, there was a gain of function. Here, it was a loss of function. So really, it was a plain curiosity in the lab. I said, we have never worked on a suppressor gene. Why don't we do something in a suppressor gene? Sean Tvetian, who was the first author of the paper, was somebody I knew. I called him up. He said, sure, we'll give you the DNA. We got the gene. And this is one of the first experiments we did. It was actually Mark Chapman, Chris Larson, Heinz Ruffner, a bunch of people together that started. But it was really Mark who I convinced to do a very silly experiment, so to speak, at that time. Some of you may remember that in many genes, we didn't know what was the transcriptional activation domain. And the way we did those experiments, you took portions of the gene, linked it to GAL4, which then binded to DNA, and then it activated the gene downstream. In this case, could be CAT assay or luciferase. So what he did was to then we had the gene, he took various components of the gene, put it in this assay, and you can see this molecule here, this fragment of 1528 to 1863 has the highest activity. These are called CAT assays. Some of you will know it, but it doesn't matter. It was just a marker. And the amazing thing is, Nature said, we want this paper, but we said, that's someplace I talked. They said, but I said, that's all the data we have. He said, but this is a very important piece of data. So this paper is 1 page. This is the whole paper. They said, this is only one figure in it. Well, of course, I wasn't going to say no. So he said, so that's how we got into it, the idea that there is a transcriptional activation domain. And the reason it was particularly interesting was that the women who had mutations in this area around these things had no activity. Only the wild type had it, but the women where the genome mutations have been shown didn't have it. So that gave some credence to it that maybe transcriptional activation domain was important, because when you had a mutation that is found in this pathology in the case of the tumors, did not have that activity. So based on that, we begin to ask, first question now is, what really is the function of BRCA1? This gene has been now out for almost 24 years ago. We still actually don't quite know what is the precise function of this gene product, except we know it is involved in DNA damage repair. That probably is one of the most important, but that's the least known. It's involved in cell cycle control. It's involved in transcription regulation, ubiquitination, centrosome function, defective X inactivation, and cell proliferation, which is particularly interesting because as a rule of thumb, most anti-oncogenes, once you remove them, the cells should actually start proliferating, right? Because they normally do is to prevent cells to proliferate. But this gene is remarkable that when you knocked it out, actually it blocked cell proliferation, and we'll come back to that one. So this is how the rendition of the three-dimensional structure that I know of looks like. It has a domain here called the ring domain, which is involved in ubiquitin ligase activity. It has a DNA binding domain, but kind of a very loosey-goosey, AT-rich domain, really. It has a transcriptional activation domain, and it has a BRCT domain, which is usually known to bind to other phosphoproteins. Many of them are DNA damage repair and DNA replication proteins. It always functions in association with another protein called BARD1. BARD1 is a protein which not much is known about it, maybe involved in splicing, but it has a ubiquitin ligase domain. Only the two of them come together, they make this region, and that's only they can do ubiquitin ligase activity. And I'll show you that in a moment. So that's all what we know. The other thing we know about it is that this gene is actually necessary during development. And our story starts a little bit like that. One day, Gerald Pau, who is hopefully someplace in the audience, came and he said, you know, Ed Leon in Rusty's lab just told me that they were doing microarray analysis and they find high expression of the BRCA1 in the day 9 embryo of the mouse in the brain, neurostem cells. And we said, well, that doesn't make sense about it. But that was the case. And then he showed me the data that, look, Phil Leader had published a paper also shown that there was high expression of the BRCA1 using the antisense in those days. So I sort of said, fine, but we are interested in transcription, right? But nevertheless, Gerald went ahead together with Chuan, who is here today, whose work is really, these are the two people whose work is all I'm going to present today to you. They got together, and I have no clue why they decided that they're going to knock out BRCA1 in neuronal stem cells, because the observation from Rusty's lab and the other one was that they were present in the neuronal stem cells. So they went through these enormous gyrations to make this mutant mouse. This is actually a very interesting one. Once I saw it, what they were doing, I got excited. But up until that point, it was really all their stuff, because BRCA1 has about 14 exons. The exon 11 is the largest. You knock it out, it's a hypomorph. The mice die at about day 9. But if you need to make one, you have to kill almost all the exons, then they die in about day 4 or 5. So they made finally a mouse which had a BRCA1 hypomorph, but a complete flux of BRCA, which we got from Anton Burns, and a nestin Cree. So that is to say, only in the neuronal stem cells in the brain this gene will be knocked out. And now they ask the question, what happens to these mice? Remember, BRCA1 knockout mice die at day 9 embryogenesis. BRCA1 full knockout die at day 5. So the question is, what happens if you have a floxed neuronal stem cell? What happens to these mice? Well, the mice were born, and you can see cerebellum is knocked out, thymus is fine. They were somewhat smaller in size, but they were otherwise okay. But they die at day 20. And the interesting thing was, when we looked at these mice, they had a very interesting phenotype. They were ataxic. They were lack of coordination, as you can see. They have tremors. They have increased agitation. And eventually, they will all die around between day 12 to 22 after birth. So this was a surprise. What does BRCA1, which is involved in causing breast cancers, has got to do with the neuronal stem cell and to do with such activity in the case of the brain? And now this is why, when this work, I assure you, could have never been done any place except Salk Institute, because we have so many neurobiologists. So the next thing we did was to start talking to everybody around, or they start talking to around, and start asking questions what it might mean. The first thing was, we noticed that the cortex is very small, 1/3 the size in the knockout. When they looked at its more histology, they found, of course, you can see there's no cerebellum, pretty much there's no foliation over here, dental gyrus is gone. This is a massive. In fact, all the neuroscientists at Salk Institute working on different mutants have something wrong in this particular mouse. And I was disappointed, because what we want to know is what happens to breast cancer. And none of this really, despite Gerald was all excited, John was all excited, I wasn't terribly excited. Well, beginning to get excited a little bit, because first thing they showed that if you knocked out, the reason the brain was small was because there were apoptosis was much increased, that's why they were small. But this is the figure. One day, David Livingston, who is expert in BRCA1, happened to come by the lab. And some of you who know David, you know how ebullient he is about everything. He's excited. He looked at it, he said, wow, he said, look, 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 this is it. You got it. And I said, what do you mean I got it? He said, look, here is the cortex, which has 7 layers of it, and Gerald had already figured that out, apparently, but I didn't know that. And they're very well marked. You see all the pyramidal cells, they're going in the right direction. It's extremely well differentiated. But when you look at the knockout, it's a mess. And those of us who grew up in cancer biology know that lack of differentiation is one of the major markers of cancer. So we said, well, look at it. In fact, this is mess. Maybe you can learn something from it, because this might be a clue to start thinking how cells might be organizing themselves with this lack of differentiation due to loss of BRCA1. So the first thing we did was to ask the question, looked under the microscope, and see how do those, the really original idea was to look at the apoptosis, how that looked like. But we were very surprised to see that, in fact, BRCA1 knockout in the neuronal stem cells had lost heterochromatic centers. There was only about 5-to-1 ratio compared to control. You can see that over here. They're a little bit more fluffy ones, but there are very few in it. So I'm building a story to tell you that how completely unrelated events begin to come, and if you wait long enough to see that there is a real story will come out of it, that we were just collecting facts. Fact number 1, it's completely undifferentiated. Fact number 2, that we have a very high levels of expression of the substantial reduction in the heterochromatic center. That, of course, made us go to the books and begin to ask the question, what are the heterochromatic centers? Well, we knew a little bit. You know the genome has euchromatic and the heterochromatic domain. The euchromatic is the one which makes all your genes, and the heterochromatin usually is the one which prevents transcription. So, in fact, they are the minor and major satellite bands which are in the pericentric region of the heterochromatin. It's SUVAR39, which is actually originally came from Drosophila, is a methylase which methylates histone at its K9, which is the lysine 9 residue. Once it is methylated, it binds to the chromodomain of HP1. HP1 is the major protein that is present in the nucleus, and it's the heterochromatin protein called HP1. And heterochromatin formation then suppresses the expression of satellite RNA. So imagine satellite RNA has to be de-repressed, because if it's turned on, it's a mess. The cell will never grow. The organism doesn't really flourish, at least not mammalians. Majority of species, yeast is slightly different. So they are suppressing the expression of satellite DNA by virtue of the fact that the HP1 is methylated and now there is no expression. Now remember, we said that there is a reduction in the amount of the heterochromatic centers here in the loss of BRCA1. So the first surprise came here. Now look at the high expression of major and minor satellites where the BRCA1 is knocked out as compared to the control. So suddenly all genes, by and large, are extremely well controlled. Even these imprinting genes are very well controlled. Here it's about 3 times more. But not the line elements, not the sign elements, all others are not increased, only the satellite RNA. And remember again, these satellite RNAs are controlled by the heterochromatin, and in the absence of BRCA1, this is a tremendously high expression of these RNAs. So that was new, and we began to ask the question, what does that mean? Well, first question was, do they bind to this? Does BRCA1 actually bind to satellite RNA? Because remember, we are saying only in the absence of it this is happening. The answer is yes, it binds to DNA. But more interestingly, it binds to the same domains of the DNA where the HP1 protein binds. Remember, HP1 actually comes in 3 forms, alpha, gamma, beta, very similar, but they are the major protein in the nucleus. And we found they bound to the same ones. So that obviously asks the question, what happens to HP1? Now, HP1, defined as a function, is heterochromatin formation, transcriptional silencing, centromere, telomere, DNA replication, DNA response. And if you remember my earlier slide, many of the same functions that were attributed to BRCA1. So we began to think maybe there is a link here now and ask the question, what happens to HP1? Once again, in the loss of loss of BRCA1, we see far fewer heterochromatic protein 1 as compared to the control. So now the plot thickens. We have already shown that there is a high level of expression because the heterochromatic centers are fewer. HP1, which should bind to, is also reduced in amount. And now the question is how they might all actually affect eventually, leading to ask us the question of the breast cancer. So here you can again see the HP1 is reduced. That's just. Now, I show you this experiment only because the reviewer said, the work is entirely in vivo. There's nothing shown here in cell lines. It was an East person, I presume. Pathetic as we are to please the reviewers, we knocked out BRCA1 in HeLa cells, and they were very happy, because once again we could show there's a reduced HP1. Then we saw these two papers, which said, role of histone H2A ubiquitination in poly-A silencing, and poly-A group protein ubiquitin histone H2A heritable gene silencing, meaning that there is a ubiquitination of H2A. And that immediately clicked, because the first thing we asked was, what about ubiquitinated H2A? And there were far fewer ubiquitinated H2A. So there's a reduction in histone H2A also in the BRCA1 knockout. Now remember, I said BRCA1 had a ubiquitin ligase activity together with the ring domain of the BARD1. Here we are talking of the ubiquitination of H2A. So years ago, Tony Hunter and my lab had done an interesting experiment where we showed that if you have BRCA1 and BARD1, only together they had the ability to ubiquitinate H2A. So another point came together now, that the ubiquitin ligase activity of BRCA1 has the ability to ubiquitinate H2A. And remember, if the H2A is ubiquitinated, it has the ability to silencing of the heterochromatin. So we asked the question, what happens if there are mutations in ubiquitination domain? And many women with mutations have this domain mutated, many sequences. One of them is cysteine, for example. So we asked the question, what happens if there's a mutation in the domain where there's ubiquitin ligase activity and that has led to the disease, meaning there's a phenotype where there's a different deficiency of BRCA1, what happens to that? Well, there's a cell line called HCC, which is made from a patient who has a mutation deletion in BRCA1. You see the high expression of satellite RNA. If we put BRCA1 back by lentiviral vector, you see there's no expression of the satellite RNA. The patient who has a mutation in the ring domain has a very high levels of expression of the satellite RNA. The others are polymorphs. So that again sinks in that something is happening here with the ubiquitination of the H2A. Well, then we ask the question by chip analysis, does ubiquitin H2A actually bind to the major satellite RNA? And the answer is ubiquitin H2A does in wild type. When the knockout, there's absolutely no binding to the major or to the minor satellite RNA. So again, to recapitulate, HP1 protein is involved in it. Ubiquitination of H2A, and the H2A can bind, and that can cause the ubiquitination of H2A. Now the problem is, ubiquitin ligase activity is actually quite promiscuous. There are many, many targets for the ubiquitin ligase. How can we say that it really is the H2A that is the target? And here Gerald did a really clever experiment, because this is as close to genetics as we can come. So Gerald took histone H2A and found that at position 119, he could remove it and put one molecule of ubiquitin. So he made a cDNA where the H2A was ubiquitinated. So in other words, you should not need BRCA1 now, because if the job of BRCA1 was to just ubiquitinate H2A, in its absence it should function. So he showed then that if you have a wild type, you have the ubiquitin H2A, no expression of the satellite RNA. But now if you take our nestin Cree mutated ones where the FLOX1, BRCA1 was knocked out, you see high expression of satellite RNA. But if you now put this ubiquitin H2A here, you come back almost near to the normal levels. I think that's as good a genetic experiment as we could do to prove that the role of BRCA1 in the ubiquitination of H2A is that the BRCA1 is the ubiquitin ligase for the H2A. Of course, as I mentioned to you before, one of the things that BRCA1 does is affects cell growth. In its absence, the cells don't proliferate. And you can see here the ubiquitin wild type, if you knock out BRCA1, much less. If you put BRCA1 back, you have once again this ubiquitin H2A can do the same job in the absence of BRCA1. Same amount of practically the replication of the cells. So we think that we know now that the role of BRCA1 is to ubiquitin H2A so that it can prevent the expression of the genes from the satellite RNA, from the chromatin, that the satellite RNA is not expressed. Well, all this so far we told you was the neuronal stem cell from the early experiments of removing them from the stem cells. Actually, in the end, BRCA1 causes breast cancer. So we did the same experiment, now made breast cancer using MMTV and so on, the other ways to do these things. And once again, when you knock out BRCA1, even in the mammary tissue, you have high expression of the satellite RNA. Of course, the real thing is, what about humans? If our hypothesis is correct that the loss of BRCA1 can lead to high expression of satellite RNA, what about women who have actually breast cancer? One of the hard things in La Jolla, San Diego, to working is really with breast cancer, BRCA1. We almost have no tissues. Practically, we couldn't get ever any samples. So we went to we had connection with the Netherlands Cancer Institute, and we got samples from women who have, so these are the tumors, which has BRCA1 knockout. You see the high expression of satellite, and here is the control. That is to say, samples from the women on normal breast in the same patient. And the reason you see sometimes much less here, it's because when the surgery was performed, a lot more of the normal tissue was left behind as compared to the control, but all of them have, these are very high expression of satellite RNA. So now we were pretty happy about the fact that we have really a good system where we started with the mouse, and we can go on to show that in the mouse breast tissue, and that is presumably the case in the case of human. And now really comes the question, how do satellite RNAs function? How does it cause breast cancer? What role does it have in breast cancer? Can we actually show breast cancer with them, and can eventually have some interesting ideas for therapeutics? So the first thing was, this is now Chuan's work, that they're present all over the chromosome, not restricted to one chromosome. So the first thing was, how does it affect? How do satellite RNAs actually affect a cell? And the first thing we noticed was they cause genomic instability, mitotic catastrophe. If you now take cells and transduce them with a lentivector, which makes the satellite RNA, these satellite RNAs are 80 rich regions. They're about 1,100 nucleotides or so. If you now put them into the cell, normally in mitosis you see very nicely mitotic spindle, and here you can see the tubulin and the actin are not separated. In fact, if you look at these cells, here is, for example, centrin, which is present on a single chromosome, and once again with SIR into BRCA1, you have a mess over here. The cells are pretty messed up. In fact, they're so messed up that if you now look at under the scope, look at the chromosomes, here is the normal human mammary epithelial cell, and here is the human satellite RNA put into them, and you see the amount of polyploidy. It's a mess. This is what is causing the genomic instability. So what satellite RNA is doing, it is introducing genomic instability, making this kind of a mess, and therefore this leads to eventually to breast cancer. Of course, this was also found now in other cancers, and sometimes there's very high expression of these genes. So the real question is now, we know this, what's the mechanism? How does it do it? How can satellite RNA actually cause breast cancer? Well, the first thing was to ask the question, what happens if you overexpress satellite RNA? And the first thing you see, there's a DNA damage response, which is by H2AX, and you see nothing in the control. This was immediately criticized, because if you put our lentiviral vector into the cells, but of course you don't know what else a mess you're making, because the virus is getting into the cell, getting into the DNA, integrating. Maybe this is the one which is causing all kinds of problems. So what Chuan did was to then synthesize RNA in vitro and then put it by nuclear injection directly, so no chromatin, no transcription, no RNA polymerase. And once again, she also found when she put satellite RNA, she showed the DNA damage here. Or she could actually do just RNA transfection, and once again you see the DNA damage. So we are convinced it has nothing to do with chromatin. It had nothing to do with actually that the virus went in there. It is merely the presence of satellite RNA that have the ability to cause DNA damage, which will then eventually lead to genomic instability, polyploidy, and to cancer. we just wanted to make, for next question, whether the effect of satellite RNA is on DNA replication or DNA replication stress. In other words, does it affect when the DNA is dividing, or it is affecting when you start the DNA synthesis, slow down, then restart it? Is it a stress at the replication, or is it stress? And this, again, is not so terribly important, except to say that the drugs which are involved in DNA synthesis do not have any difference with the scrambled RNA or satellite, same amount of H2AX. But if you now use hydroxyurea, hydroxyurea is the one which causes replication stress, because you take over the ribonucleotide reductase and so on. And when you look at that, now, in the absence of that, you see hardly any gamma H2AX. So we know it's at the level of DNA replication stress. Then Chuan did another interesting experiment. All the time we are introducing RNA from outside, or the virus from the outside, or RNA transcription from the outside. She did a clever experiment. At that time, Patrick had just come to the Salk Patrick Sue, and the idea came of this Cas9 DNA, which is inactivated. So it can bind, it can function as to bind, but it can't actually do anything by itself because it can't do the nicking activity. So he linked, we got Cas9, linked it to VP64, which has the ability to activate transcription, and now transfected into the cell. So there's an endogenous. So we are not adding anything from outside. We are merely activating the endogenous the synthesis of the satellite RNA. When we do that, once again, we have the DNA, quite a bit of DNA damage. So then the real experiment to do is, well, if we can show that the satellite RNA is the one which is responsible for making DNA damage, making the tumors, can we actually not make a tumor just with satellite RNA rather than with BRCA1? So then Chuan made a lentiviral vector where she just expressed the satellite RNA. We always have to have SHP53, otherwise the cells will just die. Because, and actually majority of the 60-70% of the BRCA1 have P53 mutation. So she then injected it in the nipple of the mouse directly, and this is really quite amazing. So all we have done is to simply introduce a satellite RNA expression in the mouse. And what you see is now about, in control we have 1 out of 13. RAS never makes a tumor, it's just a control here, and there's 16 out of 27, just injection of the satellite RNA. That is a pretty impressive experiment to think about. All we have done is really satellite RNA in the breast, and they are making the tumors. These tumors are pretty much like the breast cancer tumor. They don't have always, some of them are HER2 positive. They should be HER2 negative on principle. Now we have found in the literature there are a number of places where the breast cancer, the triple negative, so-called triple negative, are also actually HER2. And when you do all the sequencing of the RNA and so on, they have many of the characteristics typical of a BRCA1 tumor. So then we ask the question, what happens if we now take cells which are making breast cancer, put just the satellite RNA, they actually make them highly, so you can see the tumors, they make them highly metastatic. And in fact, these RNAs are able to make the primary tumors, metastatic tumors, as well as then go on to lungs and other places. So clearly, satellite RNA alone have the ability to cause a tumor which has the same properties as that of a BRCA1 tumor. So now you can think how far we have gone from knocking it out from the brain to come to a point where we can actually begin to induce a tumor with the RNA, satellite RNA, which has never been shown before. So now comes the question, how does this satellite RNA actually cause the tumor? It is not just by itself going into a cell and doing the tumor, it is doing something else. So Chuan then asked the question, what are the proteins that are bound to satellite RNA? Are these the proteins which are actually involved eventually in causing the tumor? So she used this method whereby, looking for RNA binding proteins is a mess. There have been hundreds of papers where lots of mess, but this is a very clever technology where you can use a MST-GFP, you can turn it on, the satellite RNA in this case, and then we can bind the proteins and purify them by binding to GFP. So the satellite RNA only turned on upon induction, to which the satellite RNA then can bind to the proteins, and these proteins were then analyzed at our facility at the Salk in proteomics to ask the question, what are the proteins that are binding to the satellite RNA, and could we learn from that? And here we hit a bonanza, because what are the proteins that bind to satellite RNA? Almost all the proteins that are involved in BCR-regulated network. BRCA1 is involved in DNA damage repair. That was one of the properties of it. And it turns out, when we looked at the satellite RNAs, many of the proteins that are bind to this satellite RNAs are the ones which are known to be present in DNA replication stress and DNA replication, MCM, PCNA. We have laminins. You can look at your favorite proteins over there. So that's, you could argue that just, you have some proteins bound to it, but how do they really do it? How do they really function? So here are the proteins all bound to it. And the first thing, if you put now just a satellite RNA, input proteins, at least this is now for the aficionados who actually want to see this kind of a data, you can see the proteins that are bound to it. But if you now add in this case, so you have the proteins bound to it. If you now add GFP antibodies, you see these proteins, MCM4, lamin1, lamin A, PRPA, they're all bound to it. But in the control, there's none of these proteins bound to it. So we now know that many of these proteins, and there are many more of them here, MCM4, MCRAT51, all the ones which have had some formation involvement in DNA damage repair. So now you ask the question, can you actually mechanistically show they actually are involved in DNA damage repair, and how are they functional? So what Chuan did then is, this is an assay called the DNA fiber assay. You take a single piece of DNA actually. You have to really show that they are single piece, because sometimes you can have fragments of it, and label them with IDU, so they're green in color. If you now add hydroxyapatite to it, they stop replication, and then you remove hydroxyapatite, wash it off, add the nucleotides back to it, and in this case you put the red nucleotide so you can start the synthesis again. So you can start the synthesis, stop this, add this, and see. Now you can see what happens when you have the satellite RNA added to it, either the human or the mouse. The mouse is called major minor, the human, they have different names, one of them called CFXR17. So now all we are doing is to ask the question, in this single fiber assays, where does the satellite RNA actually affect in DNA replication stress? Because we would argue that if this is functioning at this stress, that this will be slowed down. These are complicated experiments, but they require a lot of number of counting, and we are talking here now of hundreds of molecules. This is what Chuan did, 400, 500 molecules, not just 10, 15 molecules, counted the size of it. So here, already before, even in the control, in the case of satellite RNA, the green is less because the replication is slow. But after you treat them with hydroxyapatite, you see it's considerably lower with both of them. And if you now look at after replication stress, once again, this is the control. And now when you put back the satellite RNA to them after hydroxyapatite, you see they're much slower. Meaning there is actually a role of these BRCA1 of the satellite RNA in preventing the replication to start. They slow down. And during that time when the replication is less is where lots of genomic instability and genomic mutations occur, and that's what then eventually leads to the role of why BRCA1 is involved in DNA replication stress. If you put BRCA1 back to it, they come back. So it clearly is the loss of BRCA1, because when we put BRCA1 back to these and then do the same analysis, they come back to it. So we think these data suggest that DNA replication in satellite RNA expressing cells was either delayed or the folks progressed more slowly after restart. Importantly, overexpression of BRCA1 could overcome these DNA replication defects. So that's a pretty good proof how these satellite RNAs are working by taking away proteins which are necessary for the replication to start after the replication stress. Other way to show the same thing is what is called the isolating the proteins on nascent DNA. So we should be able to find these proteins on the nascent DNA. So you label them with EDU, and once again you can see that when we have the control, you see all these proteins are substantially reduced in the presence of the satellite RNA. You don't see them. So they're just not bound anymore to this. Now, what will be really nice if we could go to a human patient who has a BRCA1 mutation, take the tumor, and show in the tumor directly that, look, now when we look at the proteins, that there are far fewer proteins in the DNA replication or DNA stress in the patient. That's very hard to do. So the second best we could do is to take HCC1937 cell, which came from a patient where there's a mutation in BRCA1. They have many of the properties of a BRCA1 tumor, and when we did that into those cases, we see that there's hardly any of these PCNA4 protein. When we put wild type BRCA1 back, the proteins come back. So that's the closest we can show that is as good as in the humans. So all this then leads us to state that the satellite RNA expression impaired the function of BRCA1-associated proteins that protect DNA replication forks and prevent DNA damage. This defect may directly lead to chromosome instability and the emergence of chromosome with deletion structural changes observed in BRCA1 deficient cells. While we were pretty much finishing this, we had Susan Glaser from Friedrich Mischer Institute. Actually, she came because she's on your board. And we said, why don't you come give a seminar? And she had published this paper that histone H3K9 methylation is dispensable for cynorhabditis development, but suppresses RNA-DNA hybrid-associated repeat stability. I didn't even know that there was actually BRCA1 in cynorhabditis, but it is. And here they see that satellite repeat expression, R-loop formation, and loss of fertility arising from the loss, and that's the second paper what she talked about. So suddenly, it appeared that in addition, then there was a paper from Julie Ellinger, which I saw when I was in England, and she has also very similar results. So it appears suddenly there's a second way of thinking about how these RNA, satellite RNA might function. In addition to taking away proteins which are necessary for DNA replication stress and DNA replication, they may also be functioning by making an R loop. And the way you do that is you have an RNA which forms during the DNA replication fork. The RNA loop can form to it, and therefore it can prevent the replication, and therefore you can slow down and cause genomic damage. In their case, of course, in the case of development of the embryos, in our case, in case of making tumors with the DNA replication. So then Chuan asked the question, how can we show maybe in addition to binding to protein, this RNA is also acting as an R loop? Now, there's only one antibody in the whole world, I'm told. It's called 9,6 antibody, which can pick up an RNA-DNA hybrid, any RNA-DNA hybrid. And what we saw that when we put satellite RNA, you see the antibody picking it up. In the presence of Scamble, it did not pick up. So clearly, an R loop is being formed. And then you ask the question, what about DNA damage? Only when you have the satellite RNA, you see the DNA damage as compared to when you have the ribonuclease H, which can remove the RNA-DNA hybrid. Finally, we can show that in the DNA replication defect in satellite RNA expressing cells is reduced. So here you have 45% S phase in this case. If you add satellite RNA, they go down. If you put scrambled, and you have 37% back to it. But if you now use ribonuclease H, which has the ability to remove the RNA from the RNA-DNA hybrid, you should go back to the original, and that's what happened. It goes back pretty much high as compared to this one. So all this then finally leads us to say that heterochromatization is a conserved genomic defense mechanism, that we have satellite RNA replication, DNA damage, and they all lead to genomic instability, which is basically what we think is happening, that you have BRCA1 needed normally. When it is not there, these proteins cannot bind. They can lead to all kinds of changes into it, both at the level of either RNA being making a complex, or simply at the level of making a loss of these proteins. And that's what then leads to cancer, and that's how BRCA1 will function. Now this, of course, immediately gives you many things to think about. First thing is, can you actually reduce the satellite RNA by making some kind of an RNA oligonucleotide and take it away from the system? Second, most women, when they have a mutation, they get mastectomy and oophorectomy, but there's no really knowledge whether any of those mutations will eventually always cause the disease. There are hundreds of mutations which actually may not cause the disease, at least not in the younger people. So maybe we can show that there's high expression of satellite RNA maybe in the earlier phases of the screening. That may be another biological test to see whether it is actually going to cause the disease or not. Finally, there really is no treatment for BRCA1. Cisplatin works a little bit, and now there's an opalaroviv which can affect the PARP, the polyadenosine ribonuclease polymerase. But that's just still in the clinic, but that's a possibility. But I think thinking about the idea of making an RNA-DNA hybrid by using oligonucleotide may be another way to make locked oligonucleotides to think about it. There are still two big questions that I have to pose, which probably people will ask at the end of my talk. First, if BRCA1 is a tumor suppressor, why do cells stop proliferating in BRCA1 cells? They should actually be going overjoyedly, getting more and more, but that's not the case. I'll be happy to answer that in the end, because some of the work of Sachin doing, we think the YAP hippo pathway is involved in that. And second question, which is obvious for all anti-oncogenes, why is it that BRCA1 knockout on all cells but only causes breast cancer? Why doesn't it cause lung cancer? It's also removed from there. Why doesn't it cause other kinds of cancer? Why is there selectivity? Why does RB only cause retinoblastoma when it's actually lost every place? Why do NF2 causes certain tumors when it's lost every place? So where is the specificity? Why does BRCA1 loss actually causes breast cancer, not something else? And again, I have suggestions for that, but we don't have any proof of it. So I end my two talks, yesterday starting with the suppressor genes, today with the dominant genes, and today with suppressor genes. And I think about it, 45 years that my career spans, 43 years of it, we have come a very, very, very long way. When I started, people actually didn't really often think, they often use cancer as a one gene, as a one disease. We now know that all cancers are different. That's a big lesson we have learned. When I started, actually, there was really no hint that the genes were the one most responsible for causing cancer. In fact, Peyton Rouse, who got his Nobel Prize, in his lecture in 1966 said, one thing is for certain, genes have little to do with cancer. And here we are, that's the fundamental basis of what we do. And second, at that time we had not realized, and when I started, that cancer is a multi-step disease. Many steps have to come together. So with all the knowledge that we have now, and I showed you yesterday hallmarks, I think it is fair to say that cancer is fast becoming a chronic disease. Treatments are a combination of therapies that include standard practices, surgery, radiation, immunotherapy. Of course, this is the hottest game in town, and maybe I'll tell you a little about tomorrow, because lentiviral vectors are the one which is using for many of these CAR-T technologies. And that all these technologies coming together actually has begun to make an enormous difference. Susan Sontag said, when she got her tumor, overlaid with mystification and a triumphant mutation, charged with the fantasy of inescapable fatality, a scandalous subject. And I think in 2017, we can say quite confidently, me or my colleagues, Jeff and others standing here will tell you, the mystification is not a black box. The triumphant mutation has been exposed and cancer is in retreat. We see new ways by which to confront this inescapable fatality, and there is every reason for poetry for 15 million cancer survivors in USA. This is astounding. If you think about it, I bet more of you people know people who have survived of cancer than dying of cancer. And that has changed in the last 10, 15 years from all the knowledge that has been gained by the science that many, many thousands of us, including many at the Salk, has participated. And one can, and this is really, for me, ending my career, it's very exciting. I started out simply making reverse transcriptase. And here we are. Although I have not cured the disease, but I am a part of this community where actually they have made a difference. And that's really the best you can ask in your career. We have a great job, guys. We have a hobby for which we are paid. We usually go to interesting places. We meet interesting people. And you almost always clap at the end of the talk, which you probably will do. With that, I'd like to thank you again. And tomorrow is my last talk on gene therapy if you're still interested. And once again, I'm delighted that I had the opportunity to work at the Salk Institute. Thank you very much. Yes. Jeff. 2 questions. Oh, what a surprise. Yeah, this was the obvious thought, right? Because we have never seen anything. And in fact, the results from everybody and everything is still contentious whether the hormones have anything to do in this case in terms of why the cells, why it is specific for the breast cancer. That's not clear. We did one experiment, actually, Gerald and John, I think Gerald, I forgot which one did it. We had a different idea. We think it could be that breast has an alternative, that every other cell has an alternative gene which can complement the function of BRCA1, which is missing in the breast. So we took HC1937 cells, you give them radiation, the cells die. Then we made a cDNA library of 21,000 cDNAs from the original GNF with the Sumit Chanda. And they put it on each one of them and find 4 clones which could completely substitute the function of BRCA1. One of them we actually characterized further called Zymed10, which actually was directly involved in the binding to the replication stress. We actually showed very nicely in that case a chicken foot coming down on the microscope. But what we haven't been able to do and haven't really pursued is that are those the genes which are present in all tissues that are missing in there. But that's one idea of start thinking. Maybe there are the same true for RB and other things. Because RB has nothing to do with the hormones, and yet it's the same problem.
And so the second question then gets back to the very beginning of your talk, in your first slide, the definition of that transactivation domain. So everything you talked about was kind of the lack of BRCA1 and the consequences of that. So is there also a role of the transactivation?
We don't know, but one thing we do know, it is binding to the BRCT domain is the same domain. And that's the one which is binding to all the phosphorylated forms of the DNA damage binding proteins over there. We haven't pursued it. Part of it was there was a very interesting set of paper called coupled transcription repair from Beverly. Do you remember her name, Gerald? From North Carolina? Yeah, it turned out to be all wrong. So we sort of didn't pursue it any further. But we haven't really followed that very much, other than I think the BRC repeat is the important one.
If you get BRCA1, if you can make the protein but it can't assemble in those complexes, or it doesn't assemble well, is the phenotype the same as a BRCA1 now?
If we take a BRCA1 null cells, put into them BRCA1, will it function like a wild type, you mean? And the answer is yes. We can reconstitute everything by putting wild type BRCA1, wherever there's a deletion. You know, we haven't actually done the actual assembly, what is bound to it. I think that should be done. It'll be a nice experiment to do it. We haven't done that.
Yeah.
Caroline.
So I'm just wondering whether actually, a much more general thing is this repliposome stalling through many different ways, and R-loops are a big part of that when transcription scrambles something. And you have a mess in the genome, and then the replication stalls.
Right, at the R-loop.
And that's when you have the R-loops and that. So that may be very general, and depending on where those stalls occur, you then may then get the different processes, the different cancer So it may come down to that as a very generic mechanism.
It's
More
the-
damage for the whole thing. So if you look in other cancers, do you then see misregulation of these heterochromatic type
I-
transposons?
Yeah, I think maybe the person behind you can answer that question. I think it is true in the case of pancreatic, the work done from Dan Haber's lab. They see that. They actually see almost 50% high expression of the satellite on those cases, but none of them have done anything in terms of showing whether any heterochromatic RNA directly. They know lots of satellite RNA turned on, large amount of it, but they haven't actually done any mechanistic work on that. So I don't know that. Is that right, John?
Yeah, I think one of your slides showed that this defect specifically occurred during S phase. So that's a fact. So that's kind of like at least gave pretty strong evidence to show that the evidence, I mean the damage response happens
It's true.
in S phase. And also whatever, how much, no matter how much of efficiency we're overexpressing the satellite RNAs, the percentage of the satellite RNA caused the damage response is about 15%, which is consistent with that's that's a percentage of the S-phase population.
At that given time. Ron?
You know, I was trying to think about why it's linked to breast cancer, but not other cancers. You said you have a theory about that, but estrogen does have a trophic effect on breast, as androgens have a trophic effect on prostate. And wondering if, getting back to sort of Jeff's point, if there isn't an underlying trophic drive in that tissue relative to BRCA1 and other tissues where it's not going to be ER positive.
I think, no, I think there's a point to be made. We haven't gotten any evidence. There was a paper from Eva Lee, actually, which said that, which was then very quickly taken down because it didn't really able to show that. That was the assumption, that that was what was doing, that there's a trophic factor because of the estrogen. So it didn't really go very far with it. I'm not saying that is not the case, but that has not yet been proven.
And if you could manipulate the satellite RNA, would that alter the course of the cancer?
Well, that's what we're trying to do, right? We want to make locked RNAs, because these satellite RNAs are 80 rich regions. They are not like a very specific oligonucleotide. It's not going to be easy because they are just 80. For us to make a control was a pain. How to make a control from 80 rich? We had to bioinformatically design it through CRIS to be able to do that. But that's one of the thinking. Jerry's already shaking his head. Maybe it won't work. It's hard, but I think we did talk a little bit to people in the Ionis, and there was some interest in it, so we can at least try in vitro first, where we have very high levels of satellite RNAs in these HCC193 cells, that we put it back and see if we can reduce some of the functions on DNA binding and so on. It's worth trying it.
You know, there's, I think that Jeff Rosenfeld said that satellite DNA has a lot of retinoic acid receptor binding sites. So it might be worth looking at that.
Could be. We haven't done that. Jeff, last question.
Yeah, I want to get back to something Juan Carlos brought up yesterday, and it really was the force of my question. It wasn't a cell autonomous effect of estrogen. It was rather a microenvironment effect of estrogen. And so the question is, suppose you take a BRCA1 mutant cell that's already been initiated in the breast, and now you transplant that to another organ that's non-estrogen responsive. Would that new environment suppress the development of that cancer? Because remember, it's breast and ovarian. It isn't just breast.
I don't have the experiment, but I can only think about what Dan Haber and other people, they have shown the expression in other epithelial tumors, which do not have direct bearing on any of the hormonal effects. But I don't know, the reason I'm reluctant to say, because I don't know if they have gone that far to prove it, except they have simply high levels of expression of satellite RNAs.
I think we always have to think about the microenvironment.
I agree. I'm not, and breast has a major domain of the microenvironment. I agree completely. Yeah. Well, thank you again, everyone. Thank you.